So, welcome back. I've got a couple ground rules, or just reminding you of the ground rules. Um, so last semester, my big and only rule was respect yourself, respect others, respect me, my time, your time. Um, what I didn't explicitly say first semester, but just became an issue over time. Whilst I am talking up here, I would very much prefer you did not have headphones in. That way I can ensure that you are getting the information that you need. I let you listen to music whenever we are working. That's not a big deal. I don't mind that. And if you have free time after you finish your exit ticket, you can get your phone out. I don't care. Just don't use my time. There's that. I don't know where my clicky thing went. There it is. I prefer that you write physical notes in this class. So I have extra notebooks up here if you do not have one. If you are one of those diehard, I only write notes on a laptop, you would be the only class I have a notebook for. I don't want to do it. That's fine. I prefer that you at least have something physical next to you because we do draw a lot. That way you have like a physical notebook to like, or something to write on that you can reference, or if you're just really good at Google Draw, that's okay too. I just ask that you stay on task. Yes, we're gonna jump right into it, y'all. So you can get your notebooks out for me, please. This semester is all about DNA. So we're if you came from another class from first semester, some of this might be review. I didn't necessarily get the memo on the order in which things go, but it's fun stuff. It's some of my favorite stuff. In fact, tomorrow is my favorite lesson in all of biology. Yes. You want one? Okay. Anybody else? All right, there you go. So, what am I thinking of? Where's my brain? It's not in my head. Oh, masks too. Make sure they're over your nose. COVID's not fun. I can tell you from experience. So yeah, yes. I have no clue. I just put my stuff away when they tell me to put my stuff away. But yeah. Hopefully spring break. But who knows? So DNA. It stands for deoxyribonucleic acid. We're gonna take a little bit of a different approach. Usually I write notes out, but we're gonna try PowerPoint for a little bit. But I will always have little doodles to do. We are going, instead of me just giving you the information about DNA, we are going to learn about DNA in the same steps that the world learned about DNA. So we're gonna kind of work in a timeline. We're gonna talk about some scientists, all that fun stuff. Before I get started, does anybody have any questions about me, class procedures, all that fun stuff? Everything's up on Canvas. I use modules, including videos. So there's a video module. You just click on it, and if you miss a class, hello. Thank you very much. If you miss a class, then you can just go on Canvas and figure it out. Not figure it out, but find it. If we do anything on paper, turn-in bins are over there. If you have any questions, you can email me literally anytime. I hate having notifications, so I tend to respond really quickly. I think that's it. Yes. Okay. So it is 1910. That's where we're starting off. It's 1910. We know that kids look a lot like their parents, their biological parents anyway. They don't always look identical. My parents had 3 biological children, me, my little sister, my little brother. I'll talk about them a lot when we get to genetics, just because we're a pretty easy example. So we all do not look alike, but we look like our parents, and that was the same back in 1910. And we know that there is a mathematical pattern to which traits show up. We'll learn about Mendelian genetics. How many of you have worked with Punnett squares before? Okay, so we know that Punnett squares work. So my mom has brown eyes, my dad has green eyes, me and my sister both have brown eyes, my little brother has green eyes. So um, there's a mathematical reason why that is the case. So I'm not screaming down the hallway. Whew. So we can infer from the fact that kids look like their parents and that there's a mathematical trait or mathematical trend that something is passed from parent to offspring. We know because this is the DNA unit that thing is DNA, but in 1910 we didn't know that. So we want to know what this molecule is. The main two contenders were DNA and proteins. So we thought proteins were passed down, and that's why we look like our parents, or we thought it was DNA. But it could have been something else. So this is the way the world found out about DNA. All right. Okay. So it is 1928. There is this guy named Frederick Griffith. I promise you that all of the scientists we talk about in this unit will not be old white men, but in the beginning is a couple old white men. So, this guy, Frederick Griffith, he was playing around with mice and bacteria. So, he had two strains of the same species of bacteria. So, both bacteria are types of the pneumoniae bacteria, but one of them caused pneumonia in mice and one of them didn't. He called the one that caused pneumonia the S strain because it grew in smooth colonies. And he called the harmless one the R strain because it grew in rough colonies. He wanted to know, why do only some bacteria make people sick? That was what he set out to find out. Around the 1930s, it's like, wait, no, my timeline's wrong, sorry. You want to know why bacteria make people sick. So strep is another good example. So we have a lot of streptococcus bacteria on our bodies, on our skin, right? It's everywhere. Not a lot of it's harmful, but if you get a certain strain, it gets in the back of your throat. It's not a fun time. Gotta go to the doctor, get some amoxicillin, and then you're all right. So why do only some bacteria make people sick? And he was studying this with these pneumonia-causing bacteria. The S strain hurt mice, and the R strain was completely fine. Okay. Also, as we move to PowerPoints, I do struggle pacing a little bit. It's something I'm working on, but if I go too fast, just let me know, because I don't have me writing to slow me down. So if I keep going too fast, just yell at me, hey, Ms. Carter, slow down. I didn't catch anything you just said. I absolutely love biology, so I do not mind explaining it twice. So yeah, S strain, R strain, Frederick Griffith, 1928. Okay, so his experiment, right? So he took the S strain bacteria, injected it into a mouse, and it died. Right? It caused pneumonia, mouse died. He injected the R bacteria into a live mouse. Mouse lived, didn't get pneumonia, surviving and thriving. He then took the harmful bacteria, the S bacteria, and he heated it up, probably over a Bunsen burner, just burned it, right? So it killed all the bacteria. He took this dead S bacteria, injected it into a mouse, and the mouse lived. Right? So live S bacteria killed it. Live R bacteria, it was alive. Heat killed S bacteria, it was alive. But then he did some mixing, and he took the two types of bacteria The heat killed S and the R bacteria, both of which the mouse lived through. He mixed them together, and the mouse died. When he mixed the two safe bacteria together, the mouse still got pneumonia. Why do you think that is? There's no wrong answers. Well, there's no answers that I will ever judge you for. It's how we learn. Why do you think that if you mixed the two safe ones together, the mouse still died? Don't worry about being wrong. We'll be okay. You've seen me mess up plenty of times. Yeah. You think that the S bacteria and the R bacteria reproduced. Love it. Anybody else? It's a good thought. All right. So, what was happening is that these live R bacteria were transforming into S bacteria. So something in the dead S bacteria got taken up into the R bacteria, and the R bacteria became S bacteria. And whatever was in that mixture that made the R bacteria transform into S bacteria, Griffith called the transforming principle. So now the race was on to figure out what exactly this transforming principle was. And that is where Oswald Avery comes in. It is now 1944. He's been working on this for over 10 years, trying to figure out what this transforming principle was. Lucky for us, we don't have to learn about any new bacteria. He used the exact same stuff that Frederick Griffith did. So, same cells as Griffith. The big takeaway here are these last 3 points. We want to know what the transforming principle is. So what Avery did is he grew these R bacteria in extract from the S cells. So likely what he did to get this extract is like take a tiny needle and suck out the guts from a harmful bacteria, or he could have put them in a tube and dissolved all the nuclear envelopes and just got left with the goop. But he got an extract from these danger cells, right? And he put them on a petri dish, right? And he put that extract just like food or fertilizer on a lawn, right? And then he grew the R bacteria on that extract. And over time, those R bacteria became S bacteria. This tells him one big thing. The transforming principle is in this extract. So he can just keep making that extract and making that extract, and he knows that there is transforming principle in there. And he did 3 big tests to figure out exactly what that transforming principle was. That was the goal, anyway. Doing okay so far? Any questions? Comments? Concerns? Screams of anguish? Okay. Okay. All right. 3 big tests. The first one, qualitative. What's in it? Two big things. He was able to detect that there was, in fact, DNA in this extract, but there wasn't any proteins, which was big news at the time because the scientific world really thought that the genetic material was proteins. Because if you think about it, right, we talked about macromolecules a lot. If you think about it, DNA is a lot less complicated than a protein. Because proteins can come in so many different shapes and do so many different things. How on earth could DNA, which is one molecule, do all of the work that all of these proteins did? So the fact that there were no proteins in this extract baffles people. So, in this extract, there was DNA, there were no proteins, which supports the idea, or if the hypothesis, or supports the hypothesis that DNA is the transforming principle, but that's not enough. We have to keep doing tests. So the next one he did was a chemical analysis. All you need is this little sentence right here. So nucleic acids, right, of our macromolecules, we had carbon, hydrogen, oxygen, nitrogen, and phosphorus. Those 5 elements made up our nucleic acids. So what Oswald did was take 4 different samples of this extract, just 4 different extracts, and he ran an analysis to see what percent of that extract was nitrogen and what percent was phosphorus, and then he did a ratio. So dividing the percent nitrogen by the percent phosphorus. They were all pretty similar. 1.66, 1.75, 1.69, 1.58. They're all in the same neighborhood. Nothing too crazy. Now, scientists knew what DNA was. They didn't know it was the genetic material, but they knew what DNA was. So we had our known values for DNA, or how much nitrogen and how much phosphorus tends to exist in a molecule of DNA, and pretty similar. Not exact, but pretty darn close. What's this tell us? What can we deduce from the fact that the ratio of nitrogen to phosphorus in our samples is really close to the ratio of nitrogen and phosphorus in DNA? That it's DNA, that we have yet another test that supports the idea that DNA is our transforming principle. But we're not done yet. Scientists have to be sure. So he did one more. An enzyme test. So he took these extracts and he added enzymes to the extract and then observed what happened. Now, enzymes are what? Which one of our macromolecules are enzymes? Proteins. Enzymes are proteins, right? So our enzymes come in handy. They do a lot of stuff. And a lot of them break stuff down. Like there's a special enzyme called lactase. Remember, ACE is enzyme. Special enzyme called lactase, whose main job is to break down lactose. So what Oswald Avery did was he took some enzymes that break down proteins. He mixed it up in that extract. He grew some cells on a plate, and they were still transformed. Does this mean, or based on just this test, can proteins be the transforming principle? Yeah, 50-50 shot of getting the answer. No. Right? If there are no proteins in a serum, there is no way that protein can be the transforming principle. He did the same thing for RNA. We're going to get real close and personal with RNA this semester. Took RNA, the enzyme to break down RNA rather, put it in the serum, mixed up the serum, plated it, and the cells still transformed. Does this mean that RNA is the transforming principle? No. And finally, he took an enzyme for DNA breakdown. Injected that enzyme in the serum. Enzyme munched up all the DNA. There's no DNA in the serum. He grew the cells, and they were not transformed. Does this support or reject the idea that DNA is the transforming principle? Support. Love it. Everybody understand where we are so far? Okay. So, we got another guy. It is 1950. 6 years later, this guy named Erwin Chargaff. Chargaff has used the information that Oswald Avery discovered and said, okay, we know that DNA was the transforming principle. I got to learn a little bit more about DNA. So he took DNA from a bunch of different organisms. I'm not sure which specific ones he used, but they could have literally been anything. Like, if you took the DNA from one of you, if you took the DNA from a pineapple, if you took the DNA from a bug, if you took the DNA from a polar bear, all living things have DNA. If you took DNA from any of them, all this stuff's going to be true. These same 4 bases were always present. And these are going to come up a lot. Adenine, thymine, guanine, and cytosine. They are what we're going to call nitrogenous bases because they're made of nitrogen. Adenine, thymine, guanine, and cytosine. Every single molecule of DNA on Earth is made up of these 4 nitrogenous bases: A, T, G, and C. Now, the proportions differed in different organisms. The amount of adenine in a mouse is probably different than the amount of adenine in an elephant, but they were always there. And what was really interesting is that the proportions of adenine to thymine were always the same, and the proportions of guanine to cytosine were always the same. So if an organism had 12.5% adenine, they always had 12.5% guanine, or thymine, sorry, adenine, thymine. If an organism had 15% guanine, they had 15% cytosine, period, end of story. They are always paired together. Adenine and thymine, guanine and cytosine. Unless something has gone terribly wrong, you'll never find it any other way. I mean, you might see it like this, but it's all the same thing. This will come in handy. It's a secret tool that will come in handy later. It's going to come up a lot. So, adenine is always with thymine. Guanine is always with cytosine. ATGC. I think at some point in time I had a t-shirt that was the ACDC logo, but it was ATGC. I was very proud of it. So, any questions? Okay. A little bit more information on our nitrogenous bases. Here's what they look like. There are two different types of bases, purines and pyrimidines. The purines have double rings, just how their chemical formulas wind up, and the pyrimidines have single rings. If you notice, comparing our double rings and our single rings, our base pairs are always one purine and one pyrimidine. There's always going to be a 2 ring with a 1 ring. It's never going to be two 2 rings. That would mess us all up. You don't have to be able to identify which of the bases are purines and which ones are pyrimidines. I do have a mnemonic device for it. A and G. Ag is the chemical formula for silver. So pure greens, you always want pure silver. So I don't know. If it helps, it helps, but you don't need to know that information. Okay, next one. This is a lot of information. I'll tell you which ones you need. Alfred Hershey and Martha Chase. So they were a pair of scientists who really wanted to know what the what genetic material is. Like I said, is it DNA or is it proteins? What is passed from parent to offspring to help or to transmit our genetics? So they studied viruses that infected bacteria. Those bacteria infecting viruses are called bacteriophages. You might also hear it bacteriophages, but I like bacteriophages. They are viruses that infect bacteria. So, all the viruses, any virus really, is DNA with a protein coat. That's all it is. Just some genetic material wrapped up in some proteins. That's what a virus is. So we can look at DNA, and we can look at proteins, and we can try to see which one of them is the genetic material. We needed a simple way to tell them apart without having to do big chemical analyses. So what they found out is that the proteins in these bacteriophages contain sulfur, but don't contain phosphorus. And DNA contains phosphorus, but doesn't contain sulfur. So they can tell whether or not a substance is DNA or if a substance is proteins based on whether or not they have sulfur or phosphorus in them. If there's phosphorus, it's DNA. If it's proteins, or if it's sulfur, it's proteins. So what they did is they used radioactive isotopes to track them. They grew some phages on radioactive sulfur, so they knew if a substance was radioactive, it automatically contained proteins. And they grew some phages on radioactive phosphorus, so if a substance contained phosphorus, it was DNA. That makes sense? That was the methods, or the general outline, I guess, of their experiment. This is 1952, by the way. So, in the grand scheme of life, not too long ago. So, Whole goal is whether or not the genetic material is DNA or proteins. So here's what they did. We're going to focus on experiment 1. They carried out 2 experiments. They took some of these viruses and they put a sulfur-labeled protein coat on them. They grew these viruses on radioactive sulfur. So now we know that if something in experiment 1 is radioactive, that it is proteins. What they did was they took these sulfur-labeled viruses and they took some bacteria and they mixed them together, let them marinate for a little bit, and they literally put them in a blender. Like went to the department store, bought a blender, took it to the lab, and hit puree. Very, very famous experiment. Chuck these viruses in a blender. So then they took their final mixture, a suspension, had a little test tube, and they spun it in a centrifuge, just whipped it around. And when something spins in a centrifuge, all of the heavy stuff sits at the bottom. So they took this little bit of cells down at the bottom of the extract, and there was no sulfur found in our cells, or found in the cells. They did it again, but instead of labeling the proteins, they labeled the DNA. Did the same thing, mixed the viruses and the bacteria together, stuck them in a clean blender, right? Spun it down, got their cells, and they found phosphorus inside the cells. So there was no sulfur, but there was phosphorus. Phosphorus made it in. Sulfur didn't. What does this tell us? Any ideas? Did proteins make it in the cell? No. Did DNA make it in the cell? Yes. Which means that the genetic material that viruses inject into cells to have the viruses reproduce is DNA. This tells us that DNA is the genetic material, that it's not proteins. So, as of right now, as of 1952, we know that DNA is the genetic material. Griffith, Avery, Hershey, and Chase told us that DNA is the genetic material. And when Erwin Chargaff told us what DNA is made of, adenine, thymine, guanine, and cytosine. But the whole goal of this is to figure out how it works. Why on earth, how in the world, does this simple strand of A, T's, G's, and C's mean all of the genetic information to make a human or a banana or a fly? How does something so simple lead to all of the genetic variation on Earth? That's a pretty big question. So in order to answer that question, we needed to answer the next one. What's DNA shaped like? In all of these experiments, right, we just know that DNA is a substance. We know what it's made of. We don't know what it looks like. We don't have microscopes that good in 1952. We barely have microscopes that good now. So tomorrow, in one of the lessons that is most dear to my heart, we are going to learn about the shape of DNA. So, there is an exit ticket on Canvas for ya. Brand new module, brand new exit ticket. The video's a little late, but it will be up, well, after this class. In the future, if you miss a day, all the videos and maybe a couple PowerPoints will be put up in the video pages page. The first question on that exit ticket is a Google survey, so make sure you fill that one out as well. Anything else I'm forgetting? I don't think so. All right. Yes. Interesting. Let me get your name written down and I will see what I can do. All right, cool. Well, if you don't have it tomorrow, well, I'll put you on a list of people that aren't in my Canvas, but if you're not there tomorrow, I'll figure something out.